So this story goes from the beautiful Pacific Northwest to Northern Europe. And really, this is where it starts. It starts in the group of islands that are between, in the Gulf of Bothnia, which is between Finland and Sweden. And this group of islands is called the Åland Islands. It was there in the Åland Islands where in 1895, Dr. Eric Adolf von Willebrand described some bleeding patients and noted that there was familial bleeding. He was a medical student, I think, when he first went there. And then he went to practice medicine in Oslo. And there in 1925, he saw a 5-year-old girl with bleeding. And she was from the Åland Islands, and she came from a large family, which he studied. She had bleeding in both the mother's and father's family, and he found that in that family, 23 of 66 family members were affected. He termed the disorder pseudohemophilia because it had a lot of characteristics like hemophilia, except that unlike hemophilia, it affected both boys and girls. That article was later translated into many languages, and the deficiency disease was known as von Willebrand disease in honor of its discoverer. And the reason that patients with deficiency of von Willebrand factor bleed is that that protein is vital for the attachment of platelets to the blood vessel wall and the arrest of hemorrhage. So here, depicted in this cartoon, I show what happens when the lining layer of the blood vessels, the endothelium, is removed. So as you know, the platelets are skimming along on the periphery of the bloodstream in the cylindrical blood flow, and in that way they're able to constantly sample the blood vessel wall for defects. When they find a defect, they have ways to attach to that surface. Is very sticky for the platelets, and when the platelets see a region that doesn't have endothelium, they stick to it and they roll on it. And the initial sticking is the binding of a platelet receptor called the glycoprotein 1b95 complex to von Willebrand factor, which has become affixed to that surface. Von Willebrand factor is bound to collagen. Collagen itself also has receptors for platelets, and there are the alpha-2 beta-1 and glycoprotein 6. And together, these three receptors, when they're engaged, they send activation signals into the platelet, and the platelet becomes activated and spreads out like a fried egg to cover as much of that denuded surface as possible. The activation also changes the conformation of other proteins on the platelet surface, including this, the major platelet integrin, which is called the alpha-2b beta-3 or glycoprotein 2b3a. Through the GP2b3a, once it becomes activated, it can bind soluble proteins, including fibrinogen and von Willebrand factor that's circulating. So von Willebrand factor is both on the subendothelium and in the blood. And when it does that, it allows the platelets to stick to each other. That's formation of a platelet aggregate. If this is a cut vessel, that will allow that to form a plug. The interaction between GP1b and von Willebrand factor also is induced by high fluid shear stresses. So shear stress is a force of one layer of fluid moving over the other, much like scissors. It's a shearing force, and when the blood feels a certain level of shearing force, von Willebrand factor, which is a big molecule, is able to unfold and expose sites for platelet binding. When that happens, as shown here, in a region where the blood has to accelerate to go past the stenosis, the platelets bind von Willebrand factor, become activated, and can aggregate in the fluid phase. So I think that this mechanism is behind certain clinical syndromes such as unstable angina and also transient ischemic attacks of the brain. The protein that binds von Willebrand factor is a complicated protein. It's the glycoprotein 1b95 complex. It contains several polypeptides. The deficiency of 3 of these will cause deficiency of the entire complex. But I just want to point out, for the purposes of this talk, this glycoprotein 1b alpha. Glycoprotein 1b alpha at its N-terminus, which is farthest from the platelet plasma membrane, is the longest of these polypeptides, and it has a stalk that serves much like the stalk in a Douglas fir around here to hold the ligand binding high above the forest of other proteins on the platelet surface and allow it to engage von Willebrand factor. In fact, if this stalk is shortened or removed, the platelets are able to, once they attach to a surface, if you let them settle on it, they can stick to it, but they can't stick to it for moving blood. And so this is all I'm going to say. My laboratory concentrates a lot on this protein, but that's all I'll say about it. Von Willebrand factor itself is an enormous protein. It's synthesized initially as a pre-pro protein, which has 2,813 amino acids. That includes 22 amino acids at the N-terminus of a signal peptide that allow us to be imported into the endoplasmic reticulum. It has a 742 amino acid propeptide that gets removed before it gets in the plasma, and 2,050 amino acids of the mature protein. So it has many domains, as you might imagine, and within the center of the structure it has these three triplicated A domains, which are quite vital for the function. In the A1 domain, it has a binding site for glycoprotein 1b. In the A2 domain, it has a cleavage site for an important protease that I'll talk more about, ADAMTS13. In A3, there's a site for binding collagen. There's also a site down here on C1 where it can bind the GP2B3A protein. And in the N-terminus, it has a site that binds factor VIII and stabilizes it from degradation, and that's why deficiency of von Willebrand factor also results in deficiency of factor VIII. When it's synthesized and transported, synthesized on ribosomes, transported into the endoplasmic reticulum. And in the endoplasmic reticulum, this two monomers will associate tail to tail by a disulfide bond to make a dimer. These dimers are considered the protomer, the basic building blocks of longer chains. And once those dimers get taken into the Golgi complex, they polymerize by head-to-head disulfide bonds. So essentially you have long chains of von Willebrand factor dimers that can reach enormous sizes. When that polymerization in the Golgi is catalyzed by the N-terminal propolypeptide, which then gets cleaved off, and this form is then stored into storage granules or secreted constitutively. Once secreted into the plasma, the nascently secreted form is called ultra-large von Willebrand factor, or ULVWF. There it encounters this protease, ADAMTS13, which cleaves it to smaller forms. The newly released stuff, the ULVWF, is extremely adhesive, and it becomes less adhesive when cleaved by proteolysis. So here's a von Willebrand factor agarose gel, which you probably recognize because most people in laboratory medicine have to do diagnostics for von Willebrand disease. And what I want to point out is that this band at the bottom is a dimer and has a molecular mass of about 500,000 Daltons. So every time you go up to a higher band, you add 500,000 Daltons. And in one of these, I've counted 16 bands. And therefore, this is a protein that has a molecular mass of 8 million Daltons, which would make it easily the largest protein in the blood. And there are others in disease states that have been calculated to have molecular masses above 20 million Daltons, so enormous protein. And here's the ADAMTS13 protein that degrades it. So this is a metalloprotease. It requires both calcium and zinc for its activity, and it cleaves one spot in von Willebrand factor, and this is a tyrosyl methionine bond in the von Willebrand factor A2 domain. The access to that spot requires that that domain be unfolded by shear stress or be melted in some way by chaotropic agents such as urea. And this enzyme seems to prefer this ULVWF, the big stuff, the new stuff, over the stuff that's already in the plasma. And in fact, when platelets bind to von Willebrand factor, that increases the cleavage of von Willebrand factor, so that when you have a thrombotic mechanism, you immediately enlist an antithrombotic mechanism to modulate it. And thus far, the only reason we know for ADAMTS13's existence is to cleave von Willebrand factor. So if ADAMTS13 is deficient, it leads to this disorder called thrombotic thrombocytopenic purpura, or TTP. And that clinically is characterized by low platelet counts, mechanical destruction of the red blood cells, when no other alternative explanation exists. So clinically, this is manifested in a number of ways that would make you think that there are small thrombi occurring in blood vessels, so a thrombi in small blood vessels. For example, patients with TTP can manifest mental changes, focal neurological deficits, seizures, blood and protein in the urine, fevers, abdominal pain, which may be due to pancreatitis, and EKG abnormalities up to and including myocardial infarction. And it's believed that the pathophysiology has to do with the persistence of these ultra-large forms of von Willebrand factor, or ULVWF, that in the microcirculation can clump platelets. These are also attached on the blood vessel walls and can attach platelets to them there. And this is generally caused by failure of ADAMTS13 to process the von Willebrand factor. So a few years ago, in the laboratory, we were examining the interactions of platelets with endothelial cells, and we did an experiment in which we took washed platelets, so there was no plasma in the platelets, and labeled them with fluorescent dye, and we perfused them over endothelial cells that we had activated with histamine. And what we discovered were that these structures formed that were long strings that had the platelets decorating them, beads on a string-like structure. And some of these were extremely large. In this case, we saw some that were about as large as half a centimeter long, which if that's one molecule, you can easily see that that would be the largest protein probably ever described. And in this case, we went across 3 microscope fields to take this photograph, and this one had approximately 100 platelets attached to it. We thought that this must be what's happening in TTP, and so we tested that. So if you stimulate endothelial cells in buffer, you make these string-like structures, and you can count them. If we then took on those strings and perfused buffer that contained ADAMTS13 in it, this is a recombinant ADAMTS13, they were cleared from the surface. They were just cut off and cleared, and you could count none. But if you took that recombinant ADAMTS13 and mixed it with plasma from a patient with acquired TTP, then the ADAMTS13 failed to cleave the strings off the surface. And the reason is that patients with acquired TTP have antibodies against the ADAMTS13. You can get TTP another way, and that's by being born without the enzyme. And in that case, if you take the plasma of a patient with congenital TTP and add ADAMTS13, you remove the strings from the surface. So this is kind of proof that the antibodies in TTP were able to inhibit the process of clearing these VWF strings from the surface. Now, we also found that this ULVWF, the large forms, are not only ultra-large, they're also ultra-sticky. I've already told you that that's kind of known, but nobody had ever measured it. And we did a measurement in which we can take the different forms of von Willebrand factor, attach them to beads, and using this mechanism called optical tweezers, we can approximate them to a cell that has the receptor and see how much force it takes to break bonds between GP1B and von Willebrand factor. If we took von Willebrand factor from plasma, we could not detect binding. So all of us are lucky to have von Willebrand factor in our blood that's there for when you need it, but it's not clumping your platelets all the time, giving you little strokes. If there's a way, and you do this in the lab all the time, that you can make plasma von Willebrand factor bind platelets, and that's using an antibiotic called ristucetin. When we did that, we got single bonds that were about 6.5 piconewtons. There's another agent called botricitin that's from a snake venom of a pit viper that also will induce von Willebrand factor to bind GP1B, and that gives you slightly higher bond strength. But if you take ULVWF, unprocessed from endothelial supernatants, and put it on the bead, that can spontaneously bind to GP1B, can spontaneously bind platelets or cells that express GP1B, and it binds with very high bond strength. And that bond strength happens to be the same as the bond strength of the naked domain if you just stick the naked domain on the beads. So in other words, what that told us is that probably once von Willebrand factor is processed, somehow that A1 domain that binds platelets becomes cryptic. It gets hidden and only exposed once the molecule is exposed to high shear stress or is bound to collagen. But in the stuff that gets newly secreted from an endothelial cell, that site for binding platelets is exposed. And this is one way that you can think of it. The ULVWF would be highly active VWF, and here would be the A1 domains, and this little green star would indicate that they're exposed. And after it's been exposed to plasma and atom TS13, it not only gets cleaved, there must be something else that makes it peaceful. It encrypts the A1 domains, and then this von Willebrand factor is not able to spontaneously clump platelets. After we did this study, a group in the Netherlands developed an antibody that was able to detect those gain of function A1 domains, and it's called a nanobody because it's made in llamas. And it detected elevated binding in atom TS13 deficiency in patients with TTP, and in this form of von Willebrand disease that's gain of function molecules. And the reason that they bleed is not because the von Willebrand factor clumps the platelet, is because the fact that it binds platelets makes it be cleared rapidly, and you essentially have a deficiency. So this group and others have shown that there's elevated nanobody binding in a number of diseases, including TTP, type 2 B von Willebrand disease, and clotting syndromes, antiphospholipid syndrome, the HELP syndrome that complicates pregnancy, which is also a microvascular clotting syndrome. In malaria, that's quite interesting to us. I'll talk a little bit more about that. And in many syndromes that are included here, you also have defective atom TS13 activity. And those include sepsis, LPS-treated volunteers. I don't know what kind of volunteers these were. I imagine they were probably lab employees. Disseminated intravascular coagulation, and certain diseases such as acute pancreatitis. So the point of showing this is there's a lot of places in inflammatory diseases where you get either a lot of sticky von Willebrand factor, low atom TS13 activity, or both. One disease that we looked at was sickle cell disease. And there's a lot of reasons to think that von Willebrand factor would have a role there. Now, just to refresh about the molecular basis of sickle cell disease. So this is a very interesting disease in which the entire pathology is caused by a single nucleotide change in the gene that encodes beta globin. So what that change does is that it converts a codon for glutamate and beta globin at the 6th position to a codon for valine. Glutamate is a charged amino acid. Valine is a hydrophobic amino acid. And so what happens is that this change makes a hydrophobic patch in the hemoglobin tetramer that allows hemoglobin to polymerize when it gets deoxygenated. And the result of that is that the cells will sickle, they become rigid, and it induces a lot of problems. Those problems include increased adhesiveness of the cells, their ability to activate endothelium. There's a lot of inflammation and oxidative stress that has been described in sickle cell disease, and it also induces a hypercoagulable state. Eventually, because the cells are sticky, they are able to occlude small blood vessels. That results in tissue hypoxia and infarction, and eventually organ failure. So there had been evidence before that possibly von Willebrand factor could be involved in sickle cell disease. We did a study in a flow chamber where we perfused red cells over endothelial cells, activated endothelial cells, and we found that the normal and sickle red cells could both adhere to that endothelial surface. And if we increased the shear stress or increased the flow over the surface, the cells would come off, and it seemed that it took more flow and more shear stress to get the sickle cells to come off. So the sickle cells could adhere to the endothelial surface, and it doesn't seem like this shear stress that you have here is about what you have in post-capillary venules, and it doesn't seem like much, but if that brief adhesion allows the sickle cells to linger on that surface for any period of time, it gives them more of a chance to deoxygenate and to sickle. We found, too, that adhesion could be blocked about 50% with antibodies against von Willebrand factor. So we looked in the plasma of patients to see whether or not they had ultra-large von Willebrand factor. So here in the center of this gel, so again, here are these are the big guys, these are the little guys. We take pooled normal plasma as a control. So here we add half a microliter of pooled normal plasma, 1 microliter, and all of the patient plasmas are 1/2 microliter. So the first thing that you notice is that these bands over here on the left from patients with SS disease are much more intense. The second thing is that these patients have molecules here that are much, much higher than the highest one found in normal plasma. So they have ULVWF in the plasma. Over here, these are also sickle cell patients, but these are patients that have the milder forms like sickle beta thalassemia. So from this, you know that there's high antigen concentration and big multimers. And I can say that the highest one here has a molecular mass that's about 2.5 million Daltons higher than the highest one we can detect in normal plasma. So we looked to see whether or not this stuff was indeed truly active, and we did that with this nanobody that I mentioned before, and that would detect these exposed A1 domains in von Willebrand factor. And so the principle of the way we use it is that if you take plasma, this is an ELISA assay, and you add antibody to it, you'll bind more and more antibody the more plasma you have, and you get a slope of how much you get. And this, with pooled plasma, that would be your control slope. And then you can compare how quickly the slope rises in the plasma from a patient. And so our activation factor, this is a kind of an index, a specific activity of von Willebrand factor, we get from dividing this slope by this slope. So in 13 patients that we studied from the Seattle Adult Sickle Cell Clinic, we found, and here's in this dotted line is arbitrarily the value we got from normal pooled plasma, so that's normalized to 1. You see that all of the patients, except perhaps a couple, all have high VWF concentrations in the blood. If we take the activation factor, some of them have normal levels or low levels, but some have very high levels. So if you take an antigen and you kind of multiply it by specific activity, you get an index of what's the total active von Willebrand factor, and that's what I've indicated here. And you see that in some patients, in this case, this patient on this blood draw, so every patient is depicted in a different color, had total active von Willebrand factor that was 10 times that found in normal plasma, so an enormous amount, similar to what one might find in a patient with TTP. And so we looked at a number of clinical characteristics of the patients to see what would correlate with von Willebrand factor, and one that we looked at was LDH, which of course is a surrogate for detecting hemolysis. And what we found was that there was a very good correlation between the level of the total active von Willebrand factor and the LDH level. So that would indicate the more of this von Willebrand factor that you have in the plasma, the more the red cells are having to traverse through and probably sticking on vessel wall surface and being mechanically damaged. We also looked at ADAMTS13 activity and antigen and found in this group of patients, so this group of patients was at steady state. They were coming into the clinic from the community. They were not sick in the hospital, and we found no differences between them and normals in ADAMTS13 activity and antigen. But I did have the opportunity to study 2 patients when I was in Houston, 2 children, that were admitted to the hospital in acute sickle cell crisis. And here, in the assay that we had then, you can see that, say, a TTP patient would, in this case, who was really severely deficient, we detected about 20% activity versus, of course, the 100% in control. And we found that both of these patients had low ADAMTS13 activity when they were admitted. Activity went up and eventually got back near normal by the time of their discharge. So why do they have this? And one of the things that I've mentioned about sickle cell disease is that they have a lot of oxidative stress. And the question was whether or not this could account for the increased active VWF and low ADAMTS13 in the sickle cell patients. So just a little bit about reactive oxygen species, since we talk about them all the time. And essentially, almost all of the reactive oxygen species that are biologically important come from the reduction of molecular oxygen all the way down to water. There are a number of steps, reducing steps here, and they're basically single electron additions. The first results in the production of superoxide, which is a very potent anion, which then gets reduced down to hydrogen peroxide and further to water and hydroxyl radical. All of these three are very reactive with biological molecules. In addition, this stuff, hydrogen peroxide, which you know because you use it to clean wounds, it serves as a substrate for the enzyme myeloperoxidase from neutrophils, and when H2O2 reacts with chloride, it can make hypochlorous acid, which of course is household bleach, and this is the way that bacteria can kill bugs when they invade us. So we looked at the effect of these oxidants on VWF cleavage by ADAMTS13, and the first experiment that we did was to generate superoxide by a xanthine-xanthine oxidase system. This again is another chemical reaction that occurs in the blood and can make superoxide. And what I want to just point out here, that if we had a xanthine and xanthine oxidase in a reaction and tested the activity of ADAMTS13 on a synthetic substrate, we could inhibit it, but only when both were there, and we could not prevent it by the addition of superoxide dismutase. From this, we did not know if it was the substrate, the von Willebrand factor, or the enzyme that were becoming oxidized, or perhaps it was both. And so if you look at the substrate, this is the region of von Willebrand factor A2 domain that gets cleaved by ADAMTS13. You'll see that the cleavage site, the ADAMTS13 cleaves between a tyrosine and a methionine. Both of these amino acids are prone to oxidation by HOCl, quite prone. In fact, methionine is the most reactive amino acid. And when methionine reacts with hypochlorous acid, what it produces is methionine sulfoxide by the addition of an oxygen atom to this sulfur. When tyrosine reacts with it, it actually chlorinates the tyrosine and makes 3-chlorotyrosine. So we thought that perhaps oxidation of one or both of these amino acids could prevent or slow the cleavage of von Willebrand factor by ADAMTS13. And so we looked at that by mass spectrometry. And first, we take a synthetic peptide. And you see that if we look for the oxidized species, actually we find a little bit just in the synthetic peptide, maybe oxidized by air, where you have the methionine oxidized, but almost all of it is in the unoxidized form. If we treat this with the hypochlorous acid, all of this becomes oxidized. So this is just showing you that the reaction can occur and it's prone to oxidation. And here's tandem mass spectrometry, and essentially what you do there is you get the mass of a tryptic peptide, and then you shatter this peptide into a bunch of smaller fragments, which are all these peaks, and then you look at the change in mass. So here, in the absence of hypochlorous acid, between this Y11 and Y12, we have a mass equivalent to the mass of a methionine. In the presence of HOCl, that methionine gains 16 atomic mass units, completely consistent with the formation of methionine sulfoxide. And if we then look at what effect that has on the cleavage rate of von Willebrand factor, you see that in a dose-dependent manner, HOCl will inhibit atom TS13 cleavage of von Willebrand factor. So in this case, the atom TS13 has not been exposed to the oxidant, it's just a substrate. But this is just a small peptide. And if what we found was that we could also oxidize that site in full-length von Willebrand factor, and you can see it here with MPO plus H2O2, which generates HOCl, or with HOCl by itself. But this actually required urea to be present, otherwise you wouldn't get any oxidation. So you had to unfold this thing to expose the site for oxidation. But of course, you don't have that much urea floating around in your blood that would unfold all your proteins and make it easy for an oxidant to get them. But what you do have in your blood is you have shear stress. So remember that the thing that naturally unfolds von Willebrand factor is shear stress. In the lab, we can apply shear stress in two ways. One is using a cone and plate viscometer, where we add the fluid here and we spin the cone around, and that shears the blood. Another one is to pump it around using a pump, just as it would happen in our own blood. Well, we tried this because this is easier, but it didn't work for a number of reasons. One was that all of this stuff would evaporate away and all the proteins would stick to the plate. So we went to this. And when we did that, if we just left the von Willebrand, so this is this methionine 1606, if we just left it under static conditions, unmoving, that methionine does not become oxidized in the presence of H2O2 and myeloperoxidase. But if we shear it, and this is very low shear stress, it becomes oxidized. So it's very interesting that it did it at such a low shear stress, and we thought, well, this can't be right. Well, it wasn't right. I think what actually happened is we used a roller pump, and in that roller pump you have to squeeze the tube, so all of the stuff has to go through a very narrow area. As it's going through that very narrow area, similar to a stenosis in your coronary or something, it becomes sheared. And once it becomes sheared, it opens, and it's either susceptible if there's an enzyme around to cutting, or to oxidation if there are oxidants around. And it's very interesting because, just to give a clinical equivalent of that, in patients with aortic stenosis, they quite often develop this acquired von Willebrand disease because they cut up the von Willebrand factor and lose all the high multimers, and those are the ones that you need for hemostasis. And as you might expect, if that's true that the longest multimers are the easiest ones to unfold, then you might expect that those would be the easiest ones to oxidize. And in fact, when we fractionated them into the largest multimers, intermediate, and lowest, the largest ones were the ones that had the highest fraction of the A1 domains oxidized. There's a bunch of other methionines in this A1, A2, A3 region. There's 14 here. And remember, this is the region that's got the GP1B, atom TS13, and collagen binding sites. We were able to detect 8 of them. And for all but one, so this one here, it'll get oxidized under static conditions. All the rest of them increased oxidation when you sheared them. And that, the extent to which they were oxidized, for example, this one here, 1458, which is down here, had to do with their accessible surface area. So this is just telling you how exposed is that amino acid. And when you find that it's very exposed, it's easy to oxidize. These had very low, here's the 1606 at the cleavage site, very low accessible surface area, and you see that it doesn't get oxidized unless it gets sheared. So, and here's the A2 domain, so when this gets sheared, this opens up, and it allows either cleavage or oxidation. An interesting thing that we found was we also looked at what effect oxidation has on the function of von Willebrand factor. And you can do aristocetin-induced agglutination or aggregation of platelets. And in this case, when at low concentration of aristocetin, so this is percent agglutination, we got a little bit of agglutination, but with exactly the same von Willebrand factor, exactly the same concentration, but oxidized, you get a high degree of agglutination. And it gained function even when oxidized under no shear, so it indicates that one of those that was more accessible was responsible. But the functional gain was even higher when it was oxidized under shear. And consistent with all of this, we got an increase in the binding of that nanobody when we oxidized the von Willebrand factor. And here, this is data that's hot off the press. Now, you can say this is all in vitro artifacts, so it's not easy to actually test the von Willebrand factor from a patient because you got to get it out of the blood, because there's so many other proteins there that it's extremely complicated. But we've looked at one patient so far, and this is a patient with sickle cell disease. Here are two methionines. Here's 1385, which is in the A1 domain, and 1606. This is at the A2 cleavage site. You see you have a little bit in normals, a little bit of oxidation. But in the sickle cell patient, you have quite a lot of oxidation. And I think that, in fact, this underestimates the extent of oxidation in vivo, because in vivo, if it gets oxidized, it doesn't come off the vessel wall. So it's hard to sample it in the blood. Okay, so I just wanted to show why I think this is relevant for in vivo stuff. So sitting in this room is Ying Zheng, who is a brilliant young bioengineer who knows how to make blood vessels in the laboratory. She makes them in a collagen matrix. And all she does is she makes these scaffolds and puts the endothelial cells in them. The scaffolds, they have a profile that's square, but after the endothelial cells get in there, they reshape it and make it oval and make it look like a blood vessel, as shown here. And here you see just the outline, a CD31 binding, and then the blue is the nuclei. So we took some of those vessels and activated the endothelial cells with 4-ball myristate acetate, and then, or not, in the control, and then just perfused over them fluorescently labeled platelets. So here are the vessels that haven't been activated, but they still have some stuff on the surface that the platelets bind to. They bind to this for a little while, so this is whole blood. There's atom TS13 in there. Eventually, it clears it off. So initially, they come through and they bind to whatever von Willebrand factor was there, and they clear it off. But if you activate them, and here's activation with 4-ball myristate acetate, you see this happening. And you see some massive amounts of net-like structures, which is all von Willebrand factor. Here you see that there's a vessel that's at 90 degrees, and essentially its opening gets almost completely covered by these nets of von Willebrand factor. And the interesting thing about this, so we've done experiments in flow chambers where we look at how long it takes for plasma to clear off all the von Willebrand factor off the surface. And usually it's all done within 2 minutes. So that atom TS13 comes in, cleaves, cleans it off, it's okay. In this case, after an hour, so this is not inhibited atom TS13. After an hour, there's still thrombi there. So here, for example, here's monitored for about 6 minutes, and you see that in the PMA-stimulated vessels, the fluorescence continues to accumulate over 6 minutes. And recall that I said in 2 minutes. In histamine-stimulated vessels, generally, they're clear of von Willebrand factor. And so what you end up getting is this. So here there's no platelets, but you can see stained in green are all the strands of von Willebrand factor, and it's really a kind of an incredible thing. These strands sometimes go from one wall to another, depending on how the blood flowed through there, and they can be extraordinarily long. Here's just showing them on electron microscopy. So these are kind of interlocking chains that kind of merge and diverge from each other. And you can imagine that. And here's a micrograph where Ying went from the bottom wall to the top wall and just labeled them with different colors. So at the bottom it's blue, at the top it's red. And you can see that in this particular vessel, there's von Willebrand factor throughout this stuff. And in some cases, you have these enormous strands that go for tremendous distances. So you can imagine, recall that in the sickle cell patients, the amount of ULVWF correlated with the extent of hemolysis. You can imagine that if you're a red cell and you have to get through this, you're going to be damaged by the time you come out of the other side. Also, I think it's possible that under extreme conditions as shown by that little movie, that the von Willebrand factor by itself might be able to occlude a blood vessel even without platelets. Okay, just one other thing about the enzyme being oxidized. That's also prone to oxidation. And here's just looking at enzyme activity with exposure to HOCl. And it turns out that its oxidation and loss of activity correlates with oxidation of a methionine that's crucial. It's at a met turn. So this atom TS13 belongs to a family of metalloproteases called metzinkins, and they all have a conserved met turn, and if they don't have it, they don't work. So this one gets oxidized, it kills the enzyme. And this is probably happening in some things like malaria because it's unexplained why they just drop atom TS13 activity. They don't have antibodies, but they lose atom TS13. So we're looking at this in other disorders. Acute respiratory distress syndrome, peripheral arterial disease. In acute cocaine use, it's very interesting. Cocaine will stimulate, in some vessels, massive release of von Willebrand factor. And in malaria. So last year, a paper appeared in Blood that I had an opportunity to write a commentary on and drew this little cartoon to explain what they found. So in cerebral malaria, it's known that the malaria-infected erythrocytes can occlude the cerebral blood vessels, but the protein that they bind is a protein called CD36, which is on the surface of almost all endothelium except endothelium in the brain. So everybody wonders, so what is the receptor in the brain? Well, in fact, what these investigators from London found was that in the brain of patients with cerebral malaria, what they were doing is that they're dumping a bunch of von Willebrand factor. For some reason, they're not getting rid of it. The platelets are binding, and the platelets are a rich source of CD36. And so you get occlusion both from the platelets and then from the red cells that come and stick to the platelets. So finally, I'll tell you about a way that we've thought of pacifying this von Willebrand factor in TTP and other disorders. And this idea came from homology. And homology, of course, means that you come from a common ancestor. In this case, proteins coming from a common ancestor. And proteins that are homologous to von Willebrand factor are the polymeric mucins. So those mucins that form gels, so they're the mucins that we have in our respiratory tract, in our GI tract, in the pancreas, in our saliva, etc. They have, like von Willebrand factor, at their N termini, they have the D domains through which von Willebrand factor can multimerize, and at the C termini, they have the C domains that allows von Willebrand factor to dimerize. And they make long chains. And I remembered from when I was an intern that there's this stuff called N-acetylcysteine that you can break down mucins with, and that was known since the 1960s. A guy named Scheffner published a paper in 1964. And the reason was that N-acetylcysteine has a free thiol, and it can reduce disulfide bonds in proteins. And particularly the ones in mucins seem to be particularly susceptible. So our hypothesis was that this NAC might be able to do the same thing to von Willebrand factor that it does to mucins. So, of course, we did everything in the lab first, make it easy on yourself. So with increased concentrations of NAC, we found that the multimer pattern changed of von Willebrand factor with loss of the largest multimers. The same thing happened if we took plasma and increased concentrations of N-acetylcysteine, you get lower VWF sizes. And so here I'll just show you a movie. And what we did here is we made VWF strings in endothelial cells in culture. We perfused over them. A buffer that contained N-acetylcysteine. So here are the strings. Hope you can see them. I guess, yeah, they're easily visible. They're here, and the flow is going to be from your right to the left, and that stuff that's in there has N-acetylcysteine. So you see that N-acetylcysteine comes in and it acts really in this experiment, probably not in vivo, but in this experiment, it acts very much like ADAMTS13 does to break off and shave off those ULVWF multimers. We looked at a couple of other parameters of von Willebrand factor function. Its ability to agglutinate platelets or to bind collagen. These platelets are fixed platelets, so there's nothing in the platelet that's changed, but as you treat the von Willebrand factor with NAC, its ability to agglutinate the platelets basically goes away at higher concentrations. Similarly, its ability to bind collagen is reduced. And if you take a concentration of N-acetylcysteine that does not affect, and in this case it's 2 millimolar, it doesn't affect the multimer pattern here. We found that the A1 domain, which has a crucial disulfide bond, that disulfide bond gets ruptured. And that's just shown here by an increase in a free thiol. And once that disulfide gets ruptured, that A1 domain can't bind platelets anymore. So even at concentrations where you don't see a change in the multimer pattern, the N-acetylcysteine affects von Willebrand factor function. So in mice, so now this is in vivo in mice. Certain concentrations, 0.4 milligrams per gram, we got no change in the von Willebrand factor pattern, but at double that concentration we did. But at this lower concentration, you see that you get a change in the migration of the von Willebrand factor on these gels, indicating that the intra-chain disulfides are being ruptured. So finally, we looked at kind of a model of thrombosis. And in this model of thrombosis, what it is done is that in mice that lack ADAMTS13, so it's an attempt to make a model of TTP, you can exteriorize their mesentery and you can activate the endothelial cells using calcium ionophore. And because you've injected them with fluorescent platelets, you can watch the accumulation of thrombus in their blood vessels. And here's the blood vessel before anything happened and then after. And here's the ADAMTS13 deficient after activation of the endothelial cells. You see accumulation of thrombus, and in some cases, in some vessels, we'd see complete occlusion. And eventually, over time, over 15 minutes or so, you get clearing of this. If these animals are treated with N-acetylcysteine, you get small thrombi, but you never get accumulation of big thrombi like this. And this is depicted here. This is just looking at the mean fluorescence intensity at one site in the blood vessel, and you see that it'll build up. These thrombi would then embolize, build up, so forth, until finally around 15 minutes it goes back to baseline. With N-acetylcysteine, essentially never got that high. So, to summarize, the question at the beginning was, is it our friend or our foe? Of course it's our friend, because if we don't have it, we'll bleed. But it can sometimes also be our foe. Told you about sickle cell disease, that in sickle cell you have high levels of highly active von Willebrand factor. It can appear there, and it seems to correlate with the rate of hemolysis. And this is probably because of increased endothelial activation and defective processing. This von Willebrand factor ADAMTS13 axis may play an important role in other clinical disorders besides TTP and sickle cell disease, and those include acute lung injury, malaria, cocaine abuse, and probably you can think of other ones. If there's inflammation involved in thrombosis, this is probably involved in it. And showed you some data about reactive oxygen species. They're able to oxidize the cleavage site for ADAMTS13 and prevent cleavage. They oxidize and inactivate ADAMTS13, and they enhance the function of von Willebrand factor. I think that all of this is actually an evolutionary adaptation to be able to seal off blood vessels and contain infections. And finally, we found a way that we might be able to treat some of these disorders with N-acetylcysteine, which reduces von Willebrand factor size and activity, decreases in vivo thrombosis, and since it's known as an antioxidant, it could prevent oxidation. So I just want to say that we're studying we're starting a trial in sickle cell disease to see if N-acetylcysteine will improve that disorder. So finally, the people who did the work. It wasn't me. At the Puget Sound Blood Center, our expert in she's the von Willebrand Factor genius, is Junmei Chen. She worked with Bill Hobbs, who to do the sickle cell work. Xiaoyun Fu is a chemist who is an expert in oxidation, and she did all our mass spec. We've worked here at the University of Washington with Dominic Chung in biochemistry, and Ying Zheng, who's the bioengineer who makes those beautiful blood vessels. In Toronto, we worked with Heyu Ni and Adeli Rehiman, who did the intravital microscopy for us, and this stuff all started with Prasenjit Guchait at Baylor, and we got the nanobody from Doctors DeGroot and Lenting.
It seems like an obvious question is whether ADAMTS13 can be used as a therapeutic, and the Google tells me that other people have thought that too. Can you comment on that as an approach?
Well, I guess that there's a couple of companies, Baxter being one, that are making it. And I think what they're trying to target right now is TTP. Of course, that's rare as can be, and that's really not their real target. That would not be a good business plan. I think it might have some use in stroke and in myocardial infarction, for example. And I think it's kind of an attractive drug because that is one, what you hear all the time if you review papers or abstracts or whatever for antithrombotics. Everybody always claims, oh, this will inhibit thrombosis without increasing bleeding. And almost none of that makes sense, right? Whenever you inhibit thrombosis, you increase bleeding. Now, here's one that actually might, because it's only going to be active at sites of high shear stress. And so if you really flood the system, I bet, really increasing the ratio of ADAMTS13 to VWF, I think it's a good idea.
What's the mechanism for bringing the hypochlorous acid together with the von Willebrand factor in normal circulation?
Oh, okay. Well, that's an excellent question. I think it's basically during inflammation. And so neutrophils, of course, release myeloperoxidase, and there's also a circulating concentration of hydrogen peroxide in your blood. I mean, everybody has hydrogen peroxide, believe it or not, circulating around in the blood, and so that substrate is always there. And the interesting thing about myeloperoxidase is that it binds to the endothelial surface to glycosaminoglycans. So whenever a neutrophil sticks to the endothelium and dumps stuff, you can stain MPO for a long time after that, bound to the surface. So I think that's one way. In addition, it's not just the hypochlorous acid that'll do it, I think. There's other things like hydroxyl radical that's produced by the reaction of H2O2 with iron. That's the Fenton reaction that seems to be able to oxidize those methionines as well. We still haven't explored that too much. Other things that are produced also, peroxynitrite, which when you have superoxide combining with nitric oxide, that also is able to oxidize those methionines. So I think there's a number of situations, but usually I would say it's during inflammation. So that's why we're so interested in sickle cell disease and acute respiratory distress syndrome and other things where you have a lot of inflammation going on, a lot of neutrophils dumping their granules, etc.
Related to that point, there's been a lot of interest in neutrophil DNA-related nets. I'm wondering if there's, and the pictures that you showed,
it looks like it, doesn't it?
They do, and so I'm wondering if it's known if there's interaction between von Willebrand factor and the DNA nets that are part of the active inflammatory
process. Well, that's an excellent question. It's excellent, and the answer is yes, that there seems to be, and that's early times. There's a group in Boston that's working on that, and they found von Willebrand factor associated. I think that's really interesting because the DNA is highly negatively charged, right? So it would attract, for example, it's going to it's going to bind, no doubt, myeloperoxidase. So it's just going to be coated with myeloperoxidase. The other thing is when that DNA gets broken down, as you do with xanthine oxidase system, you're generating superoxide and H2O2 and stuff like that. So I think that's an excellent question, and exactly just what you saw there, that really what we're getting is nets, or we're going to call them webs just so that we don't use the same term, but I think that's there probably is a lot of innate immunity to this system.
And do usual doses of N-acetylcysteine that are used currently, you think likely they would have an influence?
Yes. So in the mice, the dosage that was used in those mice was approximately although given more acutely, the dosage used in acetaminophen poisoning. So if we did this and we really wanted to treat someone, you'd give it intravenously and you give it for a long time. And I guess, you don't want to go overboard and give too much, but I wouldn't be too conservative is what I'd say. It'd be something like the Tylenol toxicity.